Okay, okay, it's recording now. So I want a sign of hands or thumb up or anything that you can hear me clearly. Okay, thanks, Adiola. Um, okay, Bridget, you could hear me too. I'm happy everybody can hear me clearly. All right. So we had covered this last week. I want someone, anybody, to tell me what they learned from data formats. I wasn't around, so I just wanted to give a brief overview again. So I want anybody, please. I would not pick a volunteer. What do you learn? What have you learned last week? What one or two things? One or two things I will learn so that I know what is missing now. If I can help anybody with anywhere. Listening, please. Don't we don't have much time. No, so you know. Hello. Please.
Oh yeah, guys, please let's start talking. 2B has just 1 hour with us, so I can start. Yes.
We have just 7 people on the call, by the way. That's the first red flag. 7 people in total, with 3 of us. So we have 4 people outside. Oh, okay. Adela, you're not present in that class. Okay, let's see. Bridget and Rukoya, were you around? It's not still serious. I just want to know what you. Uh, this is not good. You know why I say this is not good? Because I didn't plan going in depth, right? I did not plan going in depth at all. So I just wanted us to start or pick up from what we've learned last week, then go over it again so that we all understand. That's the plan. So uh this one that people are not here and are not discussing. Let's say let me just go ahead. So for the people that are here, the 3 people that are here, Bridget, Adiola, and Lukoya, I would like you to please follow me as much as possible. Um, Adiola was not around last week, so please, I would like you to take. So let's start now. Let's start together. Here, data formats. So this is not new to you, by the way. I'm sure you have heard of a lot of things. So in data formats, by the way, you've heard of pasta. We've heard what we talk about FASTQ files. You heard us talking about GFF, GenBank. When Abib was going through NCBI, okay. Oh, thank you. You learned about faster FASTQ system and BAM. Good, good, good. Thank you. So let's move on from there. Abib, can you mute? I think somebody is in your background that is making noise. So let's move on. So what's the data before we even discuss data formats? Data is information. Data is defined by the information it contains, and then the design and exploitation of that information. So what I'm trying to say is, I'm sure all of us here have used what's the name before? We have used everybody has used Microsoft Office Word. Everybody have used Excel before. Everybody has used Adobe like PDF. Everybody have used PowerPoint. If you're a designer, you might have used Illustrator, Photoshop before. After every file, let's take the basic and the most common that everybody uses, Microsoft Word. If you save the file, it saves as .docx, right? The way I know, everybody knows that's how we save as or .doc, depends on the version of Microsoft Office you're using. Just thumbs up if you are agree with me. I want to believe we are at least that literate. Okay, thank you. So if that is the case, and we have agreed together that we have .doc, .docx, and all of those things, then you should know that the same thing happens in bioinformatics. And okay, one thing I should discuss about those things is the reason why you have those formats is because those file formats, the extensions when you save your file, have been designed in such a way that any file that is in that format, right, the software that would open it will is optimized for that software. Like it is optimized to do the best for that software. Another example, I don't know if anyone uses Mac here or anyone uses Windows. If you're using a Mac software, right, and you open that Mac software with sorry, let's say a Mac software is .dmg, or even your WhatsApp, your Android phone. You have apps on Android phone, and somebody uses iOS. Android phone apps are .apk, right? iOS are like .ios, right? So if you open a .apk file on an iOS, it's not even going to open because the iOS will be confused like what's this. So and that's because the file formats or the way they have organized data in on what am I calling it? In the APK file that you are going to install is being designed in such a way that only the software that would run it, that's your Android OS, will be able to piece together and use it. And that's because it has been optimized for that. One another point of optimization is this. If you have tried opening PDF file in Microsoft Word before, you will know this edit. If you didn't convert it to Word, but you just open the PDF in Microsoft Office Word, you will see how everything will be disorganized. If you open that same PDF file in Adobe Reader, you will see how beautiful and pretty it's going to be. So the point I'm trying to make is that a data is the information it contains, this is the first thing, and then how that information is being designed and optimized, right? So talking about bioinformatics, we have different data formats. A lot, right? Not that much, by the way. Don't let me say a lot. But these data formats are there because they solve each problem. We could have everything .txt. You would see the beginning of bioinformatics, they all save their files as .txt. But it was a problem because when you want to do quite a number of things, TXT would not serve you for a million and one things. So you have to actually develop or design something, a format that would help you solve the problem you want to solve, right? And that's what we're going to talk about today. So I have a table that I will show you later, or a diagram that I made last year. If the only thing you pick from this stuff is that diagram, I think you are good to go. If that's the only thing you could pick, right? After I'm going to spend a lot of my time. So if you could actually understand that file format, I think you are very, very much good to go. So let's start with that. I'm back to my slide now. All right. Okay, I discussed this earlier in the time. They were using plain text, simple. So initial sequence was stored in simple TXT files, nucleosides and amino acids, and you cannot add annotations. You'll see what I mean by annotations very soon. And of course, there are limitations to that. If you know what we have now, the basic of FASTQ files, you can see what is included in FASTQ files, right? FASTQ files have risk codes and all those things inside of it. So you cannot, um, you cannot do all of those in a simple text file. That's why FASTQ files will help you do that, right? So there's limitations, and today we have different formats that help solve problems like that, right? From the simple, very similar to TXT file, that's the FASTA files, to the most complex one, GFF files, right? Genomic feature format, right? So it's very important for us to know what this are, and that's what we're discussing and covering. They were covered it last week, by the way. I shouldn't be going deeper, but I'm not happy because some people were not around last week. And if I should go and skim over, I'm sure I'm going to confuse them more. That's why I'm going deeper. I was going to skim through this part and just go into the wrong way of things. I think it might be confusing, but let's see how it goes. So this is the table I want to talk about, a diagram I want to talk about. In bioinformatics, we have different analysis we could do, right? So the analysis you will do will inform the format of file you'll be working with. There was a joke I did said he had sometime last year that what bioinformaticians do is just convert between the various file formats, which is something very, very it's intuitive if you think about it. It's actually interesting so because that's actually what we do. But of course we did just do that. We do that with an intention. So we know what we are doing. Not just converting between file formats because if you want to know a variant for a genome, you have to use a VCF file. So you have to find a way to convert your FASTQ file to a VCF file. And then you know that for you to get a VCF file, you need a reference genome. By the way, you understand what all this means later. So we have like how many arms that I have in this center circle? We have the sequence data formats, we have the alignment formats, we have the annotation formats, we have the common general formats. This is what everybody uses. We have quality control formats, right? That's also very similar to what everybody uses HTML. And we have protein data formats, right? So all of this, I think this tree covers almost every type of file format you would ever come across for using bioinformatics. It took me time to draw this last year. And now that we have it here, I think everybody, that's what I'm saying. If you could, if this is the only thing you have access to and you could skip, I think it's enough for today. But we're not going to go through everything. We'll go through most of it, right? And then we'll move on from there. Is everybody with me before we start one after the other? We have 11 people now on the call. That's good. If you're with me, just thumbs up, please. Thank you, Bridget. So, thank you, Olariwati. Yes. So, let's go on from there. The very first file format I will discuss is Gembag format. And I shouldn't start with this intuitively, but I just want to bring it up for us to discuss. It's the one of the oldest file formats that have ever been used, have ever been used. Uh, I said it, this is already one I said. And from the point I wrote here, you can see it was made to be bridge the gap between human readable representation and a one that computer can be processed. So they want something that humans and computer can actually use together. By the way, if you know anything about computer, you know that when you are trying to make something that is accessible to humans and also accessible to humans and also accessible to what's the name, to computer, you don't, it doesn't end well, right? So most of the time it's not useful for man, it's not useful for the computer. So you'll see what I mean when we start talking about graph files and the term index files. So uh JMBAR format was the first file format that was created, one of the oldest formats that was created. And the extension is .gb and .gemba. And the screen grab you can see at the right hand is very interesting. You can see it is very detailed, right? Even though the detail is also um it's coded in such a way that the computers can also understand, right? But it's very, very detailed, right? There's the locus, that's the location at which it is, what the definition, that's the name, the assertion number, the version, because if you can see, this is the reference sequence. Aside from me writing the reference sequence, let me ask a question I did at first that was 2 weeks ago. What other thing would make you know that this is the reference sequence on the locus? I want anybody, please. Aside from being writing it as a, if they didn't write that reference title under the title, or they didn't write it as a ref sec, the keywords, what other thing would make you know? I'm specifically pointing your attention to the locus that will make you know that this is a reference sequence. I didn't mention it 2 weeks ago. Anybody, please? Let me just continue. So I do like my whole teacher. I will not tell you the answer. I will just move on, and you have to go and find it yourself. Because I did say it.
Sorry, please, can you go over it again? I'm not sure I heard what you said.
Okay, the question I was asking is, if you look at the locus, right, that locus part in the screenshot I said, the image you can see on your screen, what is something very, very specific or peculiar that makes you know that this sequence is a reference sequence? Something on this screen that makes you know that this is the reference sequence. And I'm even not just, I'm telling you, look at the locus. All the answers are in front of you. That locus, something, just one character lets you know that this is the reference sequence. I mean, this we talked about it last week. You mentioned it, right? When you're
Sir,
talking to about SBI. Eh?
I think the is it NC or
So, thank you for trying, but it is the underscore. Said it now. If you have NC underscore, anything you see underscore, that underscore tells you that this is the reference sequence. I did, right? You mentioned it now. It's on the call. You mentioned
Yes,
it.
yes, 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 I did. And you are right.
Yeah,
So,
you mentioned it. I don't know why we did not, because I was in this platform, you said it. It was here that I know it, but you mentioned it, and I was like, oh, it's very good that you mentioned it. Was teaching yesterday when you mentioned it. That's not good that we forgot that. Not happy with that. So let's move on. So this is the reference sequence. You can see the details. So the point I'm making is uh, JBank format is a very, very detailed format. So you can generate it by origin. You can see that details every part is actually detailed. The sequence starts from the origin, right? So those are the five major parts of the GenBank: the locus, definition, ascension, the version, and the origin. So that's the five most important parts, right? Of the GenBank command. You can actually do it with the other ones. All those reference authors, keyword, and all those things, and all these blah blah blah. You might actually do it with it. So you can see some GenBank commands that don't have those things inside of them. Or you can see some that are detailed as what you can see on the screen at the moment. So let's talk about those things you could see. Metadata. That's the non-essential part I talked to you about. Trying to reference. They are non-essential part of the entry, including deleted or missing information, correct references, databases, all those things. They are not important, right? The machine does not need it. No matter the analysis you want to do, I'm sure you don't need this metadata, right? Except when you want to start correlating data. But even when you correct, you don't need this name of the author, blah, 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 right? You don't need this. It's the non-essential part. But you need feature, right? Describe the important part of the sequence, such as the coding sequence, this is protein. I took a screenshot, right? So that's how I could see it like this. But if you actually go, I take the screenshot of this screen, or I think I will send the slides to everybody so that I can go to this side because I can't cover everything. This side is long and it is detailed. So uh, the coding sequences, the proteins, and the primarily intended, as primarily intended, that feature section is intended for the computer processing part. So this is where you are seeing when they are bridging human, and metadata is for host human. The feature is for the computer, right? And then the sequence data, actual sequence, that's at the origin, right? It is uh it might be DNA, it might be protein. I should put that out there. So it might be DNA and protein. You know, when it's protein, you can see the origin, you can see on the right. This is DNA sequence on the bottom, but that's the protein sequence at the top of it, right? I just abruptly cut it off. So it is marked as double slash to indicate that that's the end of the entry, and that is for the computer. So you can see where they're incorporating human and computer interaction in one file format. So that's what GenBank format is for, actually. It's for us to use as humans and also for the machines to use. And as I said earlier, something that's very important about file format is that file formats like this is that it's always not. You will see something in the front in future. Uh, when I'm talking about that file format later in this conversation, you see that they made it in such a way that they want to the best you can use with GML file format is at the moment is to teach, just like I'm doing to you, most likely. If you want to use it for analysis, it will be slowed down because of the file and the weight or the size of the files. So it is not optimized for analysis. Even though your computer can tell you if you want one more in some work or one way or the other, it's not actually optimized for analysis. So I think that's what I need to talk about in GM Bank format. So the bottom line is you don't need it most of the time for your analysis. Now this is where we're going into the nitty gritty. Here we're talking about faster formats, right? We know what format. Can anybody? Yeah, I've written it on the screen already. So it is the most important, one of the most important formats in bioinformatics. I think it's the most used file format in bioinformatics actually. Faster format is one of the is the most except okay, except for FastQ. FastQ is the most popular because that's what every sequencing machine spits out. If you are using no matter the tool you are using, you mentioned it, Illumina, Nanopore, PacBio, Sanga. Everybody spits out. I think Sanga is different, by the way. I think it is quite different. But Illumina, the next generation sequencing, everybody agrees to use FastQ. And of course, you know what FastQ is. So that's why it's very popular. But most analysis, except for newer tools that are accepting FastQ reads, right? Most analysis will allow we want you to go and align your reads first and make a faster file. So up till date, most of the work you will do have been rooted in faster files. So one of the things that will make you know that faster file is a faster file is a greater than sign. It was mentioned. I'm sure they would have mentioned it to you last week. So this greater than symbol is the hallmark of a faster file. Anytime you want to know you are working with faster now, look at the top, let's say greater than figure. Once you see greater than, you know that you're working with a faster file. Can you tell me what you would see that will make you know you are working with a fast Q file? I want anybody to answer me that. I'm sure you were taught last week. For people that were in the class last week. For people that were in the class last week.
I think that's the at symbol.
Thank you. Thank you very much. At symbol would help you know what you are doing fast use file. Greater than symbol if faster files. Okay, we are moving, we are getting somewhere. We are getting somewhere. Oh boy. All right. So it starts with that, and then the sequence ID, as you can see here, the name and some amount of text. Then the line that follows it will tell you, oh, okay, this is the sequence that we're dealing with. This is the sequence, the actual sequence after you have assembled it together. That's what you get after you have removed, after um after the line that has the greater than symbol at the top. The sequence that follows is just two lines. The first one at the top, and then the next one is your sequence ID. And the file extensions of FASTA. Most of the files you might see FA. I think that's most. I've seen FASTA, I've seen FA, but those are the two most common ones. But I've seen FA more than FASTA. So any of them can be used. All software that you use to process a FASTA files recognize FA and not FASTA, so you don't have to worry about that. So nomenclature coordinate, DNA sequence length is measured in basis of base pair. You will know that. So 100 base sequence is equivalent to 100 base pair sequence. There's nothing very different. Okay, Abib, can say what you want to say.
All right, thank you. So regarding that faster file, so I just wanted to double check something that .fa and you can have .faa and .fna. So one is holding the faster file for amino acid sequences. So that one is .fae. So whenever you see .faa, so I mean, have it at the back of your mind that it is actually holding the faster file for amino acid. And anytime you have .fna, so that one is holding, I mean, the faster file for nucleic acid. So those are the two
Can you say
distinctions.
that again? I think you're repeating.
So I said sometimes we can also have .fna
FNA,
and
yes.
yes, and .faa. Double A. So one is for amino
Yes.
acid, I mean the faster file for
One,
amino acid, and one is for
and one
nucleic
is for
acid.
nucleotide.
So .fna is like for nucleic acid, faster file for
Nucleic
nucleic
acid.
acid, and .fa is faster file for amino acid. I mean,
Yeah.
and we even have .ffn for. Let's shall continue.
It's also a faster format. Yeah. So what is the versatility of faster files? As you just mentioned, you have mentioned the versatility already for the conversation we had earlier. It's not just limited to DNA sequence. You can also use it for protein sequences, right? For DNA sequence, the letters correspond to the nucleotides, and for the nucleotide sequence, for the amino acids, the amino acids actually correspond to the letters are the corresponding amino acids. So you don't have to start racking your head or start trying to put codons together. It's just straightforward. Letter A is what amino acid that it represents, and letter B what amino acid that it represents, and all the likes. So that's what happens there. Now let's move to part Q. I think everybody knows part Q. I shouldn't even wish my dad was part Q. Do we? If you thumbs up if you understand part Q. I'm not going to ask you to answer any question, don't worry. So you can thumbs up really. Not only thumbs up, okay, like what just that. Thank you. So if only one person can thumbs up, I don't even know like this. So let's move on. So I said it earlier, most next generation sequencing platforms, everybody has agreed to use FASTQ formats as their file format. By the way, that's not the first file format that's been spilled out from the machine. Different machines have different ways they spill out their reads. Then you now convert it to a FASTQ format. If you are working with Illumina, you would have the DCL file, right? I don't know what's here already. There's a DCL file that comes, and if you want it, I think you can actually access that DCL file if you want it, and you can convert the DCL file to FASTQ. So the machine, by the way, you don't have to think about it. Once you are done sequencing, the software automatically in the machine converts the DCL format into a FASTQ format. So all you have to get to your FASTQ format. If you want to do probing, I don't know why you need it, by the way. I don't kind of analyst. You can go and get the DCL file. If you are working with Nanopore, they call it pod 5. I work with for quite a lot. The file format that is determined before you pass file is the port 5 file. So that's the file that IPO reads the electrode charge and all those things that happens when your nucleotide goes through the port. And then your port 5 files are always converted to FASTQ. The same thing, packed by they have their formats. Is B back H5 and back H5, whatever it is. And then what you get after is your FASTQ files, right? So the whole point I'm trying to make is that everybody has agreed that FASTQ file is the ultimate result that everybody should get from any sequencing platform, either long read or short read, right? So FASTQ files, the anatomy of FASTQ file is it has 4 lines majorly, right? Sometimes 3, but most times it's always 4. And the fourth line, of course, thank you for the person that answered earlier. It starts with ask. So to refresh our memory, FASTA file starts with what? Edamilo.
Greater than.
Thank you. Did you die? No. Answer my question. So FASTA file starts with greater than. FASTQ file starts with ask. Those are the things you should know. This one should be at the back of your hand, at the back of your head. I don't know. You should just make sure you have it with you at all times because it will help you when you're sorting files. There's some files that might be named wrongly. I don't know, but it's good to know this thing. Now, the first file line starts with the art, and if it has started with the art like that, you should know that it's the last few file. And there's a read sequence identifier. So after, by the way, it is more complex than this. I would explain it later. I have the slide dedicated to explaining that first line. But let's go to line 2. That's actual the read sequence, the actual read sequence. Cytosine, thymine, adenine, guanine, all those things. That's in the second line. Excuse me. The third line is just a concatenation time, just a plot to tell you that this is like a splitter so that you don't get joined together. And then the fourth line is where your press calls are, right? They are ASCII characters that tells you the quality of the reads in line 2, right? Line 2 is the read, line 4 is the quality score. Do we get it? I'll explain what that means in the next slide. So the fourth line of the FASTQ entry encodes numerical values known as the press score, right? And it tells you the likelihood of the errors. So it's a very, very interesting way of interpretation. Now, the table you can see below, let me show you the, of course, you can see it already. This is how long the press score is, like from 0 to 40. The higher the press score, the better your it. And by the way, it should be intuitive to you know why we're representing press scores as character. Who can tell me why we're representing press scores as character? Just think deeply. I want you to actually think why didn't they just write it in the background that 1, 2, 3, this first one that is C, this one that is this first C here, this first C is A. Let's say A is like maybe 33, right? Why didn't they just write C 33, C 33, G 33? You get why did they choose to represent you with the character? Can anybody? It's a very simple answer. But I want us to think. Can anybody think and tell me why they chose to use press course? I just need answers in the next 5, 10 seconds. Nobody's answering me. Think now, it's not hard. Huh. Abib, are these people in class last week? Let me continue.
Well, some people were in the class, but I think quite a lot of them were not in the class.
Oh, okay.
But that
All right.
question, I
So
think probably they should be able to answer it because we've already
yeah, it
taught.
is
I taught
obvious.
something like that when we were doing, data compression.
Yeah.
So
Okay, so
I'm not sure if they can
Yeah. Somebody wants to respond. I heard a woman,
approach
the
it.
lady.
yeah,
Thank
I guess
you.
it's to save space because like
Yes.
for example 40 as like a two
Characters, yeah.
two
Exactly.
digits,
Two
I mean two
characters.
yeah,
Yes.
so
Yeah.
it's like
Yeah.
so it's best to
Adiola,
like
Thank
replace it with
you.
something that will save
Yeah.
space.
Thank you. Did you
so
die? Oh, thank you.
Adiola
Abib
and
continue.
Crickets. So I'm just clapping for them.
Okay. Thank you very much. So it's to test space very, very easy because you want the quality score to match the reads. Do you get so that you don't have extra space and you don't get confused? The machine will be no matter how smart the machine is, it's going to get confused at the same time. So you want to make sure you don't have that. So you have to represent your quality scores with an annotation. So and it is very simple. We have ASCII table, right? So ASCII is American Standard Character something. I can't remember what ASCII is at the moment. So if you know it, maybe you can write it in the chat. But that's what we have, right? So we just converted ASCII table into our quality scores. We map it with our quality scores. And the higher the quality score, the better your reads. Now, quality score is actually the interpretation is inverse of what I said, but it is also the same. Just hold on and let me go to the next slide. You understand what I mean? Yeah, American Standard Code for Information Interchange. Thank you very much. That's what it means. So it's for encoding complexity of encodings. Let me show you what I mean by that in my next slide. Good. So if you have an ASCII figure character of 0, what it actually means is this. You have 1 in 100, there's 1 in 1% chance of error. That means that read is bad. The machine was guessing and it gets wrong. And if you make it do that thing a million times, it's still going to like that score it's not confident of that score. The machine that calls that score, that base, is not confident of it at all. Now let's move to quality score of 10. If you have a quality score of 10, it means if I call this figure, if I'm to call this base 10 times, there is a likelihood of me missing it um 1 of 10 times. So I can get it right 9 times, but it's very sure that 1 of the 10 times I'll miss it wrong. But you would want to see that is actually a problem because if you are sequencing that read like 10 times or 20 times, you're sequencing it 100 times, right? That means you are going to get all out of 100 reads, 90 times to be good, but 10 of the 100 times to miss the figure, right? So you don't want something like that. By the way, that is the code. If you are using nanopore, if you are about to set up your read in on nanopore, the base quality score on nanopore is I think 9. Minimum quality score is 9. You can't decide to push it up or drop it down, depends on you. But by default it's 9. I think Illumina is around 20 something or 30 something. I don't know. So the higher your quality score, the better or the more confident you are in that rate. Now, if you go up to 20, it means if I should call this point 100 times, I am very sure that I will get it 99% of the time. So that's when I say quality score of 20, I am confident, I am very confident that that read is okay, it's better. But the higher, you can go higher. So 30 means if I should call this 1,000 times, if I should read this sample 1,000 times, I am sure this days I have given it will be what it is 999%, 999 times, right? So that's one thing I should like. So the whole idea of quality score is the higher the quality score, the better certainty. That's the word I'm looking for, certainty. The higher quality score, the better the certainty. See how 40 is very, very high. That means if you call it 10,000 times, I can only make mistake one time. I'm sure that 999% of the time, this figure I'm giving you, or this read I'm giving you, this adenine I'm giving you, or this timing I'm giving you, is going to be what I have found 999% of the time, right? 999 times. So it's very, very interesting that you could see the level of confidence that the machine is displaying. So if you want to know how good your read is, right, look at the quality score. But of course, you can't start looking at all the quality score one after the other. Okay, what's the name? The first read. You know how many reads you generate? Sometimes in a read, you generate a million an experiment. In a sequencing run, generate as much as a million reads, 2 million reads. You want to start going through each read one after the other? You can't. That's where fast QC is there, right? Fast QC is for quality control, right? I think we have discussed this before. This quarter I've learned fast QC sometimes before. Have we? Have we
No,
not?
no, did
Oh, we
not.
haven't. Okay. I'll touch on it. By the way, if you run a fast QC file, you get a result that is a report. So this quality control format, if you can see what is here. And FastQC reports, you can see it's HTML, then you can combine all your FastQC for your different experiments to make the most QC report. Excuse me. So let's go to FastQC. I discussed FastQ. Okay, I wanted to say what is on the top before I move on. Remember, I was saying this is the label, right? This label, okay, this is the summarized label because of the image, but this is how it actually comes up in all of the times. And this figure, as gibberish as it looks, is actually saying something very important. You know, we sequence on a flow cell, right? We have mentioned flow cell before. If you remember your bioinformatics, from bench to bioinformatics, from lab to bioinformatics, we've mentioned something about flow cells, right? I'm very sure we mentioned something about flow cells. And the reason why I was saying fast QC is a bit is because when we're doing bench to bioinformatics, I mentioned this fast QC. I mentioned it briefly. But let's fast forward. I'll discuss it again now. Now, after you've extracted your DNA and you have done your light preparation and you load on a flow cell, right? Either Illumina, Nano, or PagBio, you load on the flow cell. Now, the flow cell is actually a very wide, it's very small when you see it, but if you look at it microscopically, it's a very big field that DNAs come to get support, right? Depending on the technology you are going to use. If you're using nanopore, the flow cell is filled with pores. If you're using Illumina, the flow cell is filled with different lanes, right? Now, every tool that has ever been manufactured, either nanopore, Illumina, PacBio, has an ID that is unique to it. That's the first part. At this machine, there is a run ID, so the amount of runs you have done. 136 run on this machine. There is a flow cell that was used to do this run. The flow cell is called FC7, whatever. On the second lane, of that flow cell is distributed into lane. There's a tile number made 2104 tile on that lane. And then on that particular tile, there is a cluster there. If you remember, I don't know if I've discussed in bench to bioinformatics. If I'm not watching, maybe you should go back and watch it because this time I'll be making reference to that part. We mentioned something about cluster generation. If you are doing some Illumina frequency, there's cluster formation. So this is a fast queue. By the way, this top line is dependent on the tool you are going to use. If you're using nanopore, it is formatted different. I'm using Illumina now, so that's why you could see this result, and I'll explain as we continue. So on the tile number 2104, there's a coordinate. Of course, we all did mathematics, geometry, x-axis and y-axis, right? So this is the coordinates, and this is the coordinates for the cluster, the x-axis and y-axis on that particular point, right? And then cannot tell you after you've gotten the coordinates, then you cannot tell you, okay, this read is from that place, and it is it's a paired end read, it's not paired end read, and all the other gibberish that comes after. The last one is the index. Who can tell me what the index is about? What is an index? In your sequencing, what is an index? Let me make it simple. What's the barcode? Anyone, please.
So think about where you were talking about batching or multiplexing samples together.
Yeah, you are giving them the answer already. I want
So,
somebody to because
Victor, you are not permitted to talk. I know Victor will get the answer. But Victor.
anyone, Victor don't answer. Actually, that's the answer. Labels for each DNA. Barcodes are labels for each DNA. So this is the barcode for this particular sample, right? Because you know when you pull samples together, they are from different. When you put DNA together, you put DNA from different samples. So you have to label them individually so that everybody knows where they are coming from. I wouldn't go into labeling again since we have discussed this in bioinformatics, bench to bioinformatics. If you don't remember or understand this, go back there and watch that. You can come back and watch this. So I actually made a pros of what this code is. It is simple. This machine EAS did a run, and this is the run IG on a flow cell disk. And this particular read came from lane 2, tile 2104, at the position X coordinate and Y coordinate on this flow cell. And this is the first read on the paired end sequence. And the real field and X, Y, and all like so. This is the barcode sequence. So that's what that line means, right? So it is. I think the one I did earlier, this one is for Danuku. I think it's also has the way you can explain it. I didn't read, but the Illuminati is how it looks, right? Most of the time, you might not have to go deeper and deeper, but there are sometimes you, depending on the type of analysis you are doing, there's some reads that would intrigue that you have to go deeper and go to a particular flow step. Maybe you just the analysis to get from simple to complicated, and you have to start digging deep and troubleshooting different things. So it's important for you to know how to interpret these things. You don't have to cram it in your head. When the needs comes to, you can always scoop it up and read. But I'm giving you an idea of what it does and what it means rather. So this is an example of an Illumina flow cell, right? There's a flow cell. There is the ID on it. That ID you saw, that's how it looks like. And then there are lanes. Those are the lanes. You can see different lanes. By the way, this is just an example. It gets very, very complicated. You can see like 1,000 lanes, 2,000 lanes on the flow cell. It gets very, very plenty. And then the flow cell will come in, go out, and then this is where you put your DNA and everything, and then cluster generation. You can see on the lane, the cluster will be formed, and then that cluster where it is formed will be the X coordinate and the Y coordinate, that what we saw earlier. So this is just a visual representation of what I explained in the last slide. To this point, do we understand, please? Do we understand?
Yes, sir.
I want Tom. Oh, thank you. If you cannot answer me, thumbs up, please, or thumbs down, anyone. All right. Rukoya, are you with me at all?
Yes, sir.
You are not responding. I appreciate when people respond. Thank
I
you.
will have to rewatch, sir.
Eh?
I said I will have to rewatch it, sir.
Oh, okay. Okay. No problem. So, and I'll give you slides too, so I think it will help. So, back to my slide. And this is a fast key format, and we're done with that. I think so. Now, I was saying something about quality control. You can't keep reading each of the reads one after the other and be telling you what's read one out of two major reads. Position one, it is see, okay, this is good, this is good. You cannot do that. You are just going to fool yourself. And nobody can do that, by the way. But there are tools that can help us. For short reads, practice is one of the best tools that this is not the best industry standard tool to check your quality control or quality score, right? It's an HTML file. After you have there's a tool called Persuasive, but after you have run it on your reads, the result you get is an HTML then file that gives you a report for each of your samples, right? So this is a sample one, and to tell you, okay, the one on the left is the very good data because you can see most of the quality score. Look at them. You know, it's 0 to. Do you remember our quality score when we saw earlier? 0 to 40, right? Most of the quality score, the list is maybe at this end, and you can see it's just 32. And even with that, you know that 32, I've done well. Do you understand? But look at the one on the right. Most of the quality scores of one on the right are very, very, see, two. Once they have crossed that 20 threshold, I don't trust the data anymore. So the one on the right is the very bad data. The one on the left is the very good data. Do we understand this threshold, or I should explain it again? Oh, thank you, Koya. I'm happy you understand this one. Bridget, thank you. So I'm happy. Let me do a little more explanation. I hope I don't confuse you. If you know Illumina sequences by sequencing by sentences. They call it sequencing by sentences. And what it does is the sequencing keys for Illumina, they are graded, right? By cycles. So it's like you're doing PCR, right? And you know in PCR, I don't know if you know PCR. Before I start saying things that would confuse you. Okay, let's start to explain again. So PCR is when you sequence it's not sequencing. You're trying to use polymerase to make a copy of a fragment of a DNA. So you have primers that would bind at the front, that will bind at the back, and then after they are bound, you load it on the machine. So there are polymerases in your master mix. What the polymerase does is, so there are 40 cycles. Let them 40 cycles. There are three major cycles in a PCR run. There is the denaturation, annealing, and elongation. So denaturation, your DNA will denature. Then there is annealing, your primers will bind. You start putting your nucleotides in all of those things. But that thing happens in cycles. There are like 40, most people, most assays uses 40 cycles. But that's not my point. My point is if you are doing that in Illumina, Illumina does something very similar to that. Very similar to PCR, but it's high fidelity this time. So what it does is it will do that PCR run. So there are kits that will do 40 cycles, there are kits that will do 50 cycles, there are 40 cycles, 150 cycles, and 300 cycles. So uh here the kits they use here is the 40 cycle kits, right? That's where in the X axis you can see 40. So it is for every fragment, it will do PCR on that fragment 40 times, right? And then each time for each one, it gives you a read. So for the first fragment, my read is A, and I am sure that that read is A by quality score of 40. You can see even the read is 38. You can see how high that yellow line, see how high it is, right? And on and on and on. But this is an aggregate of all the threads, all the reads in that particular sample. So I wouldn't have to go deeper. But you can see that the quantity score are on the y-axis. Most of the reads in this sample, most of the reads are all in the gray. That is all you need to know that, okay, my sequencing went well, and I can use this sequence to do downstream analysis and anything I want to do. But the one on the right, if you look at it critically, it is a mess. And it is a mess because most of the reads from position 20 down, most of the reads from that particular sample from position 20 down have a quality score that is lesser than 20. That is very bad. When you target to position 32 to 40, you can see how bad their frequency is, like their quality score is. So if you have this kind of data, you cannot use it for anything. If you use this, you are just wasting your time. Victor, do you understand my point? Okay, thank you. So there is something else they call multi-QC. Now, fast QC runs on a sample individually. So if I want, I was talking something earlier about barcodes, right? About indexing. And why do we index? We index because we are multiplexing samples together. So I can sequence 24 samples at once. I can sequence 48 samples at once. I can sequence 96 samples at once. I can sequence 360. I think there is Novartis that does open 360 samples at once, right? Now I can do that because I have barcodes on each of the samples. Now, when I'm doing my fast QC will give me, if I'm doing 96 samples, my fast QC will give me 96 reports of something like this, 96 different reports. Do you want to kill me? So I ran away from reading individual lines and I was able to get fast QC that would do it for me. Now, fast QC are these results for 96 samples. I cannot still go through 96 samples. Then you will use the tool called multi-QC. Multi-QC will help you put together all the reports from fast QC into just one simple report. So it's just one single report that that will tell you that okay, sample 40 is bad, but sample 130 they are very good, their reads are good. So fast QC is not doing anything. It's just helping you aggregate what multi-QC rather. It's not doing everything. It's not doing anything. It's just helping you aggregate what fast QC has reported. It is simple as that. So these are other metrics in fast QC. I'm not going to talk about it. But multi QC, I mentioned it in the diagram. I just thought to touch it. Do we understand what I mean by multi QC before I move on to Sam and Vampires? Thank you, Bridget. Thank you, Bola Sido. Thank you. Good. I'm happy that you people are doing thumbs up. At least it gives me encouragement to continue. And if you don't understand, don't thumbs up. Just tell me you don't. Write in the comments. I will go back, right? I like when people communicate with me. Let me express it now. I like when you communicate with me. It makes me know that, okay, these people are actually listening to me or not just pressing their phone or speaking, right? That's why I want you to respond. If you understand, respond. If you don't understand, respond, right? So let's move on from there. So, the next file format I will be discussing is SAM and BAM files. And if you know SAM and BAM files, can anybody tell me what they are for? It's on the screen already, by the way, but from what you learned last week, just a line. If you know, fine. If you don't, no problem. But anybody? Just start with SAM files. Okay, thank you for alignment. Thank you, Victor. So let me take us back to the diagram I drew. So sequence data format. We have done FASTA, we have done FASTQ, we have done GMAC. We have finished this arm. That top left arm. I'm not sure if you can see my remote. A bit sorry, that's the remote. My first of all, I'm sure you cannot see it because I was sharing a screen. I'm not sharing the whole entire screen. I'm just sharing a tab. So let's move on. So the FASTA sequence, the sequence data formats, they are all in the left, right? Right? But alignment, by the way, let me continue. Let me continue. I'll come back to that later. Alignment formats. This is what you have of them here. We have SAM and BAM files. And then, what's the full meaning of SAM file? Can anybody tell me? Full meaning of SAM. Oh, yeah, Google it and tell me. I'm waiting. You cannot Google. How are you now streaming? How slow can your internet be? Anybody? What's the meaning of SAM? Hey, okay. I think who has to start buying data for these people or buy them 5G broadband because they don't even know how to use the internet fast. Monica
Sequence
Ejimi,
alignment.
thank you. Sequence alignment. So let me go back to my expire. Sequence alignment map. Thank you. Now your internet just start working. If you cannot even cheat in the exam after how many hours? So SAM is a sequence alignment map file. And what it does is simple. Let me try and take it from my head before we go through the slide. So, you have reads. Reads are like fragments. You know what I will do? Let me continue. Reads are fragments, right? Fragments of DNA. You know how we get reads, right? After you have extracted your DNA, it is almost impossible, except you are using some special techniques. It is almost impossible to get just one long strand like you have successfully extracted the whole genome. It's almost impossible. It is very likely that you would break and fragment your DNA strands, right? So in the moment of extraction, you will break your DNA strand. And then when you start your library prep, you would break your DNA strands, right? Now, those broken DNA strands are what you are going to feed to your sequencer. And at the end, you want to be sure that after you have fed them to the sequencer, they will come back together as the whole genome, right? You want to make the initial genome from the fragments. Now, how do you do that? You will use something called a reference. So when you have when you have a reference, you will now, it's like you arrange it. If you have arranged, if you people that have Ajebo childhood, that you have Lego bricks. I'm not talking to people that don't have Ajebo. Victor, I'm not talking to you. But people that have Ajebo childhood, that you played with Lego bricks, you would know that for you to actually arrange a Lego, you would have seen what the Lego set has. And you see an image of what you can build with the Lego set, right? So you now start arranging the Lego set to make what the cardboard or the paper that comes with the Lego set means, then tells you that Lego set can make. So some of you can make cars. There are Lego sets that are for cars. Even there are very, very complex Lego sets now that you can make aeroplanes. But you can only make that because you have the reference. That this Lego set can help you make that, right? So if that is the case, what you are going to do is you get, in the case of informatics, get a reference, right? And then align the reads to the reference. Do you understand me? So you would get a reference and align the reads. Maybe I will just escalator. I'm trying to stop sharing and share my entire screen. I don't want to share one tab anymore. I want to teach. I was supposed to break through this because we have a long way to go. We are going to slide 32 and we are still on slide something. So saying that if people, will just say, Morin, who at the beginning of this class. I should not have gone this deep, but let's go. Um,
Are you sure you'll still be able to finish this thing? It's already 1 hour.
Habib, it's 1 hour. It looks like your prayers are you have your prayers come to fruition now.
No, I mean, if when you are going is actually okay, you can probably continue with
Okay.
I'm going to stop sharing.
It looks like I
But
have
if you want to finish, so
I'm coming. I'm coming. Okay. I have something that I drew last year. Let me stop sharing and share my entire screen. Share entire screen. And now that I've shared my entire screen, I'll not be able to see your individual reactions. I'll come here and look at your fragments. I'll just I'll bring it up here. Then I'll make a line. Now, this one at top is my reference. This is my reference, right? I have my reference. Oh, I don't know how to spell reference. Victor influence. Oh, this is my reference genome. And then these are my reads. So I'll map my reads against the genome. So my reads will have lines to different parts, like you're arranging labels. But you are not the one doing there's software that will help you to do it, right? And after the arrangement, you get a consensus sequence. You see my slide. So everything we arrange, then you come into something. That's what I have here. Actually, why was I even doing that? So different reads will come together, and after they come together, you would see the consensus conceive, right? Do we understand what I just explained by SAM file? There are fewer more slides that explains it properly, but I want to be sure we understand it. Do we understand what I have just? Hey, Victor did not understand. There are other people aside from Victor. So let me do it again. Thank you, Olari Wajirasuga. What BAM files, I mean, what SAM files does, we'll talk about BAM files later. What SAM file does is simple. All your reads, you would need them on each other. Remember when you break, let me go back to that my escalator. Let's escalator, whiteboard. Yeah. Yeah. This was from last year. This was from the first quarter. Hey. So I don't know what I was thinking, but it was. This is my DNA sequence. This is the old genome before I extracted it. This one. But once I am done extracting, I will never, it's almost impossible for me to get this old genome back. So what I will get instead is fragmented pieces of this genome. And they would look like this. Okay, you said, will they align as a complement? You think the purpose of sample is to get the consensus? Yeah. Abib, you can come up and say, Abib, you can unmute and say something, please. Abib, take over.
yes. So I mean, regarding Victor's question, so it's asking this: would they align as a complement or based on exact nucleotide? So I mean, it's just one. Would they align as a complement or based on exact nucleotide? It's based on exact nucleotide, not as a complement. So, and if the read is coming from a reference, I mean, from a complement sequence, I mean, probably from read two, so that one is also going to be a fast. How do you put it? So it's going to be converted to the reverse sequence before they are aligned. But so there is a way, I mean, Samsung can actually probably determine whether the read is coming from the first round or from the first round. So there's a way it can determine that. But it's aligned based on, I mean, exact sequences. I'm not sure if I captured that. And okay, regarding my own comments, so I mean, what Toby is trying to like describe is what we called, I mean, Kemi mentioned it in the last class, the reference based assembly. So that is, so there are two ways that you can use to assemble your genomes and your reads. It's either you use de novo assembly or you use reference based assembly. So if they're using a reference based assembly, you have to first, I mean, you can't escape it. You have to first do alignment. So you have to first do alignment before you can now start looking at the consensus. I mean, multiplied at each positions. But at the end of the day, so a sound file will not actually hold a consensus sequence. It's not going to hold the sequence, consensus sequence. It's just going to like hold the reads. So just imagine it's going to contain information about your past three files and also the position where they actually map to your reference, but it won't contain any information on the consensus. I'm not sure probably where so we can to rephrase it better. But it doesn't hold the consensus contiguous sequence. It's just telling us information about the reads and where they are mapping to, and also their mapping quality. So yeah. So
Oh yeah, sorry, I muted. I've been talking now. So maybe I jumped myself because I explained in the data slides. This is how a sample looks like, right? It's a text type format that contains the alignment information of sequences that are mapped against reference sequence. So um if you are doing assembly, right, as Abib said, that you want to do with reference-based alignment, assembly alignment like that, you would need a reference, and that is where you need that's where stamp files come into play, by the way. So the files contains unmapped sequences and sometimes are text format, so they are more readable and human. So by the way, this is also a more readable format, and from what I said earlier about readable format files, most of them are not computer. Well, this one is actually computer friendly, but it's heavy. It's not space friendly. We'll talk about that later. So what it contains is this. It's simple here. We have the query template name, aka the read ID, right? So each individual read, you can see their ID there, and then a flag that indicates the alignment information about the read. So if it is paired, if it is aligned, the flags actually there is something about the flag that we'll talk about later. And then the reference sequence name. So that one is almost the name of reference sequence you are actually using. Then the position of the reference. So our position on the reference is that read mapping, right? And then the mapping quality. So hold on. It's a very, very so let me start a little game where the flag, right? We have the read name. The read name, that's your unique, it's a unique identifier for each of the DNA fragments. So I told you we broken fragments and we frequency fragments, and then each result that are broken have become a read. And if it is a read, there should be an ID for each of the fragment that have sequence. So the unique identifier is that our read name, that's the first one. Then the flag, like a code number that summarizes how the reads are actually aligned to each other. So it tells you if the read was paired with another read, or if it is aligned successfully, or if it is mapped in reverse directions. The paired end will paired end alignments actually have odd number values, but unpaired have even numbers. There's something about that I should talk about. We'll talk about later. And then the chromosome. So which chromosome that's the reference sequence country. If the region aligns anywhere, this will show a star. I think I have something on my slide that talks about that later. So scratch that, please. I'm coming. Then let's talk about the map quality, right? It depends. It tells you how. So something about the map quality that I'll talk about is this. If your read is mapping to a reference genome, the map quality tells you how how unique is that read mapping to that place. Now, if a read can map almost anywhere, let's say the read is 15 base pairs long, and it is filled with a lot of A's, right? Homopolymers, A. And reads like that used to struggle. Machines that reads like that used to make the machine struggle because the map quality will be low. And why is that? Because it is um 10 sequences, 10 base pairs long, and the 10 base pairs are A's, right? Map quality will be low because there are like different parts of the genome where there are plenty A's. So that read, you don't know if it is from the first part of the genome or the middle part of the genome or the end part of, let's say there are three parts of the genome that there are a lot of homopolymers, right? But the higher the mapping, the map quality, the higher that figure, the higher you know that that's actually a unique, it is unique for that place. So if the map quality is higher, right? Let's say I think it's from 0 to 10. The map quality is higher, the higher the map quality, the higher the uniqueness. So if you have a map quality of 10, you know that that's map quality. What am I calling it? The read rather is specifically for that place that is mapping. That part of the reference is where it is mapping. If you try to put it in another place, it is not going to map. So that's how you know the quality of your mapping. You will understand this point. You will understand that point that I just explained. Can anybody tell me yes or no?
Well, I think we can just continue now. So when we
Okay.
when we get to that, so when we get to sequence alignments, so we'll talk about all of this stuff a little bit further. I mean,
Okay.
because we'll still
So
be doing
there's something they call Cigar string too. So Cigar string is like a coded summary of how your alignment happened. I think I have a slide that talks about that. So maybe we should move there Seth, so that we can talk about it. The one I'm just talking before my slide like this. I haven't skipped the header, right? The same thing like we have in the FASTQ format. There's always the header. So the header is the reference sequence identification and the program identification. So the first line start with SQ, indicating the reference sequence config, right? And then the SN label identify the config name. That is chromosome 14. This one is chromosome 14. And the LN label indicates the length of the config. So how long is that config, right? So typically reference genomes include multiple configs and corresponding SQ lines. So of course, if you imagine you have mapping to a human being reference genome, we have 23 chromosomes. We have This is huge. You cannot put all of this. You know how many billion sequences we have in a genome. So it's actually interesting to know that you cannot have um you cannot have just one lane. So your reference genomes have multiple configs, and of course that will come to multiple sequence. And then the second line is PG tells the program that you use to. By the way, you will know what BWA is later when we start frequent alignment. Start doing all this alignment properly. Know what it means, right? So detailing the program that you use, the page line help you know what program is. I think it's both eye alignment, both eye alignment, right? I mean that BWA is both eye alignment.
No, Burowila alignment.
So yeah. Then multiple PG lines may appear if some files are merged. It often contains just one. So you have tags, you have IDs, you have PNs, and then the version numbers, and then the commands used to execute the alignment. You can see how detailed this is. See the PL, let's see the command. And then the VN is version number, then the PN is the program number. Very beautiful thing. So the sound files actually, it is obvious that sometimes the machine does not need this. If you want to do downstream analysis, the machine actually don't need all of this information. But because of us humans that want to pull through and know what is happening, what is aligning and what is not aligning, that's why we have the sound files. Right? We talk about BAM files later. They are very similar to sound files. Very similar. Actually, they're identical. It's just that they are binary formats. Now let's move on to what we were discussing earlier before I came here. Now, the body I was going to explain the Sega file, the Sega numbers, right? I would skip this part because if you have this, you can read it. It's detailed. It's actually very explanatory. But yeah, the Sega file, the Sega, it has it's a way of summarizing how aligned a read is, right? Now, this part is where the Sega files are. The 8M, 214M, 1D, 3M, those things are very simple. M means matches. So 8 nucleotides match. Then there are two insertions as extra bases in that are in the reads. And then there are two deletions. There are another four matches. Then there is one deletion, and then there are three matches. So this is a read, and this is how it's matched to the reference genome at that particular position. So the first line it matched the reference genome at position 7, and you can see that it has 8 matches, then 2 insertions, and 4 matches, then 1 deletion, and then 3 matches. So it's a very beautiful thing to if you understand, if you see how these things work, right? There are matches, there are addition. So it is if you see what I have down here, I wrote an example down on the left. So if this is the reference, if the reference here, see read 1, read 2, the reference matched perfectly. That's 14 matches, right? But in in the second one, we have 6 matches, 2 insertions, 1 8 matches. So what the second part means is there are 2 base present in the reach, but are not in the reference. So these are the 2 insertions, right? 6 and 8. So you can see 6 plus 8 is 14. And that was the total in the first place. First one is also 14. But there are 2 insertions here that were not there. So it is the way that, and this is the read, this and that read. So it's a way to tell how the reads map to the reference without actually having to bore you with a lot of other details, the reads mapping individually, without you looking at the nucleotide actually. Well, this helps you to see. It's very simple and straightforward. Do we understand this figure operation? Do we? Okay, thank you, Bridget. Thank you. So there are other examples. Thank you. There are other examples here. 14 matches, the match sequence perfectly. There is another one, 7 match, 3 deletions, and 4 matches. So the reference has 14, but this one has 11. 3 of the nucleotides have been deleted for this particular read. So you can see how it matches, like we can interpret these things like that just as we So we have 2 matches, 2 deletions, 4 matches, 3 deletions, and 3 matches. So it is just each of the reads will be doing that. That's how everything is doing. If you have a million reads, all of the reads will be matched like this. So you can see that, of course, it's interesting for bioinformaticians to look through and see how your reads map and enjoy it, but it is going to be voluminous. You have a million files, a million reads per sample. This is one sample. You go to sample two. Let's say you sequence like this samples together. You are going to have a million reads. Let's say, okay, it might not be a million reads for the 196. Let's say, you just can imagine how voluminous the space will be. It takes gigabytes and gigabytes of space if you have samples. But there is a way we have actually evolved to corner that. We don't use BAM files and SAM files anymore. We have BAM files. BAM files are exactly the replicates of SAM files, but they are binary coded. They are in zeros and ones, zeros and ones, and zeros and ones. So these are BAM files. So BAM files contains the same information as the SAM files, but are in binary format. So you cannot read them as human because you can't be reading BAM files. They are in ones and zeros, making them smaller and more efficient for software processing. So while SAM files are human readable, BAM files save time and reduce computational storage costs. So it's storage friendly, it's computational friendly. Then there is something they call sorting and there's something called indexing. Let me read it out so that I will then explain after. So after generating BAM files, the first step is to sort and index. So BAM files often start or sorted by read identifier. So sorting by genomic coordinates is typically necessary for downstream applications. Common tools for sorting include SAM tools and PCAT. You will learn when you start sequence alignment. I'm going to talk about these tools. They are interesting tools and you'll see how they work under the hood. So when sorting BAM files, users can choose between sorting the sequence identifier or by genomic coordinates. So most of us expect BAM files to be sorted by genomic coordinates for accurate analysis. So what happens in that kind of sorting is when you have your read, right, your reference genome, right, by the way, and you have your reads that have mapped the reference unit, right? And of course, this is not in this is not in the human readable format anymore. They are being converted into sequence. When you want to start looking for different parts of the genome that you have, you you must have so you have to sort the reads by the coordinates. So you want to see what are the reads that map to the position 1 million and 200 and something thousand this pair. Let's say there's something that you know that's important in that place. You want to see there's a mutation. All you have to do is just go by genomic coordinates, look for that position, and then you see the risk that map at that place, and you can do whatever you want to do at that position. So, importance of sorted BAM file. Many bioinformatics tools that analyze aligned reads require inputs in the form of a sorted BAM files, making sorting an essential initial step for processing. If you are doing BAM files, by the way, you start doing when we start sorting alignments, we almost run these two seamlessly together. So it's by default, it's supposed to be it's second nature for you to run the two together. Once you sort you index together at the same time. And how does a BAM index file looks like? Maybe the index file will make you understand what it is. It's typically accompanied by a BAM index file, those as by file. And what it is it serves as a table of contents of the BAM file. So by files is much smaller and of course it's just a table that is an index. There's a table of contents of the whole book. So you have a chromosome, you have reads that are mapped in binary formats in different parts of the reads. You have a table that tells at what position what reads are there. So position one, we have 10 reads that are mapped, and then this is blah 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 blah. Position 10, these are reads that are mapped, position this, on and on like that. That's how the BAM table is. So it is easy when you want to sort and you want to look for things. And what is the purpose? The BAM file indicates where specific reads or sets of reads can be found within the BAM file, facilitating efficient access to data without needing to load the entire BAM file into memory. Of course, the said BAM file is lesser than SAM file, but it's still large, by the way, considerably large. So it is important to sort. So it is by default, you should know that when you start doing BAM files, SAM file, BAM files, you have to sort. And then regeneration after sorting. Since the location of the reads in the BAM files can change after sorting, it is crucial to generate or regenerate the by file after sorting the companion BAM file. So this ensures that the index accurately reflects the structure of the BAM files. Do we understand that line? So since the location of the BAM files of the reads in the BAM files can change after sorting, it is crucial to generate a by file after sorting the BAM files. After you have sorted, So you know there is difference between sorting and indexing. So you can sort, and after you have sorted, you have to after you created your BAM file, you can create a by file, like a indexing file. But if you should sort, which is advisable, you have to create another by because they would have rearranged. So you should create another by file. So the by file can be created using tools. I've mentioned the tools I've produced before and the software requirements. And then the application is both BAM file and by files are generated and they can be utilized for various analysis, depending on what you want to do. We'll talk about what to do with them in subsequent classes, right? But I'm talking about file formats today. So this is another type of file. I think I should stop here because I have gone somewhere and I've overshoot my brother. Abib, will you take over? I will send the slides. And did we talk about was this taught last week? This year file.
I think, yeah, then we finished everything. But I think probably we should stop here. So because
Okay.
like when we actually get to all of this file, we'll still revisit them when we start. I mean, we'll revisit VCF when we get to freelance calling and also some file. So we'll go more in depth
Yeah,
there.
when we start doing the actual file, but it's just good for them
So
to know
everything, yeah,
that the,
yes, so everything will make sense then. So I mean, I think it might just everything might just be so abstract right now for you. But once you
yeah,
start working with some of these files, you actually knows that I mean they actually make sense, and there are reasons why we have to like have the captured information in that format. So yeah, we're thinking you
thank you
guys will
very much.
probably read before the class, and so that you can actually probably ask questions, so that probably to be can
That's that.
address or to read before the class. So, yeah, so you just have to like stop here. You know, we are time bound, so we have to finish on time.
Yeah,
So thank you very much,
yeah.
Toby. Yeah. So
Thank
I'm
you.
not
I
sure
was
whether
great.
you guys still have memory cell left for me to just introduce what.
So, guys, should he take? He wants to take some other things, but do you think you have the bandwidth to learn what he wants to say, or we should stay till tomorrow? They will not still talk. Is it not the people?
All
If
of
you
them
tell you
will
want
be
to
happy.
give them money, they will not talk. What
We
is
should
thumbs up?
continue.
Answer me in the chat or mute. Continue. Stop.
Please let's stop. Thank you.
You see? Require stop. Adiola stop.
No
No
bandwidth.
bandwidth, though. Austin.
Okay, Austin.
All right. Maronel,
Is
see your
you
people.
guys are like it will be so glad if you guys can actually probably read up, please. Some of these things, I mean, in fact, there are things that you can actually read yourself and actually understand
Yeah.
even
Yeah.
without us probably making the revision. But the reason we actually take it this class is because we know you guys have a lot of things you are doing. The exam is starting. You don't actually probably have a lot of time to dedicate for bioinfo. So, and we don't want to stress you a lot. So that's why we are trying to like make things simpler for you so that you don't have to like do a lot of readings and figuring out. So, so I probably I'll just continue tomorrow. I have my slide, although I was just hoping to introduce, in fact. So, but this is okay. I have 30 minutes. Okay, shall I continue? Okay, I mean, let me just show probably 2 or 3 slides so that you guys can actually probably install at least some tools before tomorrow's class. Just 3 slides. I promise you, I'm not showing any code at all. Just 3 slides about the tools. Please just wait for like 10 minutes or 15 minutes, and then we'll be done. So let me try and share my screen.
We still chill on this call. Touch is not saying anything.
I'll be in the workstation.
Okay.
Okay. So, I mean, tomorrow's class is going to be on NCBI eDirect. You know, we started with data. Please, can you see my screen?
Yes, yes,
Yes.
yes.
Yes, okay. So tomorrow's class is going to be on NCBI eDirect. And, you know, we started with databases. I mean, when we started, playing around with NCBI, also with all this MBU Uniproct, right? So it turns out that, I mean, all those things, all those activities that you guys were doing graphically, I mean, using the graphic user interface, all those things that looks pretty to you, you can also do them on the command line. So everything, probably if you actually wants to even read any article, want to read the article, let's probably want to search PubMed database. I mean, it's like you can actually do it graphically. You can also do everything on the command line. And the actually reason why you might actually want to do that, because we told you that, I mean, NCBI is a very rich repository where, I mean, you can find a lot of sequences. People deposit sequences a lot. But like as a researcher, I mean, we don't actually have the money to start, okay, saying, okay, want to isolate any organism and also wants to sequence. We don't have the money. I mean, Bioinfo does not have the money to actually probably do that or embark on such activities. So the only option left for us is to actually probably fetch data from like publicly available databases. I mean, like we said, we have scientists that do deposit their genomes or their sequences, whether genes, genomes, or anything. They do deposit, even metadata set, they deposit everything on publicly available database. And one such example is NCBI. The other one is DDBJ, and the other one is we have Pathogen Watch. We also have what again? Embu. So, and if you so just like I said, just like I mean, you can download this thing I mean, using the graphic user interface. You can also do them on the command line, and the tools that will actually allow you to do that is what we call the NCBI indirect. So this indirect is like just see it as like a suite of packages. So by the time you install this indirect itself, so you have access to a lot of packages. So there are a lot of packages I mean, the indirect suit and this suit and suit. So, what's the pronunciation of this? This, sorry, can somebody pronounce this, Stephen? This sweet. Anybody?
Sweet.
So
Sweet. Sorry, but you get the gist. So that's what I mean. Okay, sweet. So once you download eDirect, automatically you have access to a lot of other functions like, okay, let me show you. Like you have options to something we call eSearch. You have access to, so you have access to eFetch. Sorry. So I'm just making my actually. So eFetch, you also have access to eLink. You also have access to another, I mean, functions or another command called I can't remember, eLink, eFetch, eSearch. Yeah. So let me check my book. So you have access to a lot, but let's continue. So, and if I actually want to install it, so what I just want to, I'm going to share this slide on the group so that you guys can actually probably install this thing. So before tomorrow, please make sure if you are still using uh, uh, what is the name of this terminal that you guys, Git Bash, please make sure you have Ubuntu. I mean, if you have Git Bash, things might actually become so uncomfortable for you. So if you actually want to make your life very easy, so just make sure that you install another terminal. You can probably uninstall Git Bash if you don't have a lot of space on your computer. But if you have a lot of space, you can actually retain it. So, but make sure you actually find a way to install Ubuntu, please and please. So by the time you click on this link, you will just paste it. So you will actually just follow this instruction. So I'm going to send the link, or if you guys see, you can just probably copy it. Let me just put it here so that I don't have to send the slide. Yes? So please make sure you copy this link. So, and then by the time you install Ubuntu, so make sure that you've just follow all of these instructions on how to install. So just follow this instruction. So what you just have to do is probably do this. just copied everything. So let me just tell you that. So by the time you just copied everything, so everything should download. So it's still downloading. So once you are done, so copy the next command, which is this. And then after that, so just do this just to confirm, test whether your stuff is this, okay? So just to confirm whether you successfully installed. So this one is the confirmation stuff. So you can see, so everything has been, it's still working. So just follow, all you just have to do is, the installation is very simple. They've made everything very simple. You can see I now have this. So if I should press search, all of those functions, search. So you can see search minus f. So you can see I have everything installed, or you can do which research if it's going to work. Let's see. So we can say I have research, and if it is a link or e-info, which e-info. So we can say I have that one as well. So so I'm going to stop here, but I just want to, if you don't have Ubuntu already installed on your computer, make sure you install it. Those are the two tasks you have to do ahead of tomorrow's class. And also make sure that you install the eDirect packages, so suite of packages. So I can't copy the link. Okay, so I'm going to probably, Toby, I'm not sure whether you are with your system. Is Toby still on the call? So I'm not sure whether anybody can just copy this, if you are able to copy it and then paste it on the
Can you
group.
again so that I'll copy it and put on the group?
Yes. Okay. So are you able to copy it from this? Are you able to copy it here? Mayone?
Yes.
Okay, please just send me to put it on the group. So those are the two tasks. If you don't have Ubuntu already install it. And also make sure you install following these instructions. And then so we are going to have fun tomorrow. So I'm going to stop here. So see you tomorrow. Yeah, we are done. All right. Bye, everybody.
Bye, sir. Thank you, sir.